So the next speaker is Lucia Melloni. Affiliations are the Department of Neurology at NYU and the Department of Neuroscience at Max Planck Institute for Empirical Aesthetics. And the talk is titled Learning to Uncover Structure, Insights from Intracranial EEG.
So thanks, Alessandro, for inviting me and for the organizer for having me here. And also, it's what I'm going to show today is something that is a work in progress. So some of the things that I'm going to be showing probably might be rough, not finished. But I thought in picking, your brains, this amazing audience to actually get, a better story out of this. And what I'm going to be showing today is trying to understand this problem, which is how do we go from something that doesn't have pauses to something that does have pauses. And this might seem kind of like obvious to all of us who already know language and understand language. So it seems to all of us that the language really, whenever we listen to people, as if there are pauses in between the words. But in fact, there aren't. And I'm going to try to give you an example as of, if you were not to, I hope that most of you won't speak Portuguese.
Onde é que é a paragem do autocarro?
How many words there were there? One? There you go. So there were many, right? And the point there is that at some point we were all babies, and we wouldn't know actually how many words there were. So the problem was, how did we get, from there are no silences between words, which is this acoustic waveform, to really try to understand that there are words there. And it's not that we have the answer yet for this problem, but we have fairly interesting hypotheses. And one of those came from a beautiful study 20 years ago, or even more now, from Jenny Safran and Rick Aslin, where they thought, okay, maybe babies are just very smart. Maybe they just compute all the statistics, possibly in the acoustic domain. So they just track whatever was before, and they look at what is the transitional probability between what they had heard and what they will hear. Right? And if the probability is high, then they will say, okay, these two things might go together. And if the probability is low, then they would say, well, maybe these don't go together. Right? And an example of that would be a word or a phrase like pretty baby. And if you look, the probability between pre-T is actually higher than T and B and bay. Right? And maybe baby is actually doing that. And what they did 20 years ago in a beautiful seminal paper was to present babies with stimuli that look like this, for 2 seconds. In this case, they weren't actually written, but they were spoken, right? And babies just had to hear to this continuous stream for 2 minutes. And unbeknown to the babies, there were actually 4 words hidden in these materials. Something like tokibu, gikoba, gopila, and so on and so forth. And after that, they asked the babies by looking time which stimuli they would prefer, and they would present to the babies 2 things: either a word that was presented before in the stimulus material, something like gola tu, or a part word, something that was made out of one word in this case, ku and daro. And what happened was that babies that were exposed to only 2 minutes of these materials would prefer the part words, or would look at the part words longer than to the words. And the idea here is that you would look at the things that you find novel. So the words, since they had heard them, they were just boring, right? So they wouldn't look at them. And ever since, these studies on the auditory domain have been repeated with, you name it, shapes, scenes, whatever you want to have, they have been tested. What is interesting is that no matter which stimulus materials people have used, they have always found statistical learning. And they have always found statistical learning, which is actually very, very fast. The question, however, is what is the mechanism that supports this very fast form of learning? And while we know a lot behaviorally about this phenomenon, we know very little neurally about this phenomenon. And one of the claims that I had, or one of the hypotheses or postulates that I have at this point is that one of the reasons why we know nothing about the learning is because of how we have been measuring it, which is that we expose babies or adults or whoever is participant, and then we ask them offline where they had learned the stimulus. So we know a lot about retrieval, selecting the word versus the part words, or this scene versus that scene, but we know nothing about how did the brain came about to understand and learn those structures. And I thought that using the technique that Nye has developed would be actually an interesting way of understanding how does knowledge get to be there. So we're going to use the frequency tagging to try to understand what is the mechanism for learning as opposed to just the consequence of learning. And we're also going to ask when and where in the brain does statistical learning occurs. And finally, and this is probably like the part where I want to have most of your input on, is which areas would be important for this. And we have had a lot of conversations about cortical tracking, but we haven't had much conversation about hippocampal tracking of information. So how do the memory systems could actually play a role in statistical learning and in general, not just for this, but also probably for language. So the idea here was that we know that the problem with the offline measurements is that, okay, what if I forget? Or what if the consequences of learning are different from the actual process? And the idea was, in this case, to use the technique that we had developed with Nye to try to track, in this case, how would learning come about. And one of the basic ideas that I thought of is that, okay, if we take this beautiful time frequency representation to the time domain, what we know is that as the brain accrues information, whatever wave, like EEG wave or any wave, would just integrate up to a point when information cannot be integrated anymore, right? And this idea, like the mechanism for that could be a distribution model. And we thought, well, what if we take the Jenny Safran study, where we present 3 syllables at a time that compose a word, and if the babies, or in this case, adults, learn the materials, they would, in essence, integrate over those 3 things. They would just chunk. It would be a simple signature for chunking. And sure enough, so we took, this is literally Jenny Safran's stimulus material. So we present words like tu, pi, ro, bida, ku, gola, bu, pado, ti, and so on and so forth for 2 minutes, in this case, to patients with epilepsy. And I'll explain you later on why that's important. And the idea here is that this is a completely synthetic language, and you can hear it's like this.
He ro pa do ti tu he ro pa do ti bi ga ku go la bu pa do ti bi ga ku tu he ro pa.
And it goes on for 2 minutes, right? And we only ask the subjects in this case, please bear with us, listen to this for 2 minutes. And we repeat this 5 times, just to see how would this evolve over time. And what is important to note here is that what defines this word is something completely arbitrary, which is the statistics. Is the fact that 2 predicts P predicts rho, whereas in turn rho can be followed by B, go, or pa. And so the transitional probability is only 0.3. And that's all that actually is the difference. And the idea here was that hopefully the brain will track each and every of the syllables. In this case, it would be at 4 hertz because each syllable is 250 milliseconds. And if subjects have learned the words, they would have something like a 1.33 hertz if they have integrated over the 3 syllables. And we had a control stream where we had the very same syllables, but just scrambled up, right? And the idea here was that there's nothing to be tracked but the syllables. Right? And the way it goes in this case is that we either present the random sequence or the statistical learning sequence in counterbalance order. We collect EEG while, in this case, intracranial EEG while subjects are listening to these materials. And then we have the typical offline task. So we present either, like the word that was presented versus a part word, something like robida, and we ask the subject to tell us which one of those they thought was a word in the stimulus before. And we have so far data from, I'm showing you only 7 cases. We have so far 10 cases with intracranial EEG that we collect in NYU. And what is interesting there is that we have, because of the practice of the hospital, we have several cases that have bilateral coverage, first of all, but also they have extensive temporal coverage. So it's a kind of like a high-density fMRI or something like that. And what we would be looking at is, in this case, in the frequency domain, if the subjects had learned the structure, we would be looking at a peek at the word rate only for the statistical learning sequence and not for the random sequence, but a syllabic rate for both conditions. And I'm just showing you here one particular subject just to show you how reliable these responses are. So I want to make a point about that as well. So this is the control conditions, and these are all the electrodes for one particular subject. So as you see, there is a very strong peak at the 4 hertz. What is beautiful is that in the distractor condition, we see also a peak at the word frequency. And also here some harmonics that later on I will discuss. I don't think that they are harmonics, in fact. And now, this is when we have just one subject. This is when we have the 7 subjects. So this is going to be the random condition. This is the distractor condition. And now what we can do is that we can do this analysis over time. So we can track how long did it take the subjects to get this distractor. And this is what actually happens. So this is the first 2 minutes, the second 2 minutes, 6 minutes, 8, and 10. And as you see, this actually grows over time. Right? What is even more interesting, and I think this is one of the reasons why intracranial will always be an interesting technique to work with, is that if we then go into the brain, we decompose the responses that we observe. We have either electrodes that would only have, and okay, this is one particular subject, just again to show you how reliable these responses are. We would have electrodes that would only have a response at the syllabic rate. We would have these very interesting responses that only have a response at the word rate, but not at the syllabic rate. And yet other set of areas that would have responses at the syllabic rate and at the word rate. Right? So these are really only chunking responses. So there is no input to those areas. And now if we take the population, what we observe is that these are the topographies of, in this case, the syllabic rate only responses, word rate only responses, and these are syllables and word rate responses. And as you see, they are in areas, even though David don't think that, they are very, orthodox. At least I think that some of them do make sense, which is that the fact that we have mostly the word rate responses at the posterior STG and the word plus syllabic rate responses in the middle STG. Now, this is just to assure you, remember that we had the control condition, so we have none of these responses in the control condition. So this is just not, crap that we are measuring. Even more important, to the discussion that Elana and I had before, and to the question whether, because you see, babies are not paying attention to language; they just learn it, right? And I think it's important to know whether they will learning, even if they don't want to. So what we did is that we have a condition where it's explicit, and by explicit I mean I tell the participants, look, there's a structure in the material that you have to uncover. I don't tell them what it is, but I prime them to be able to show, to look for a structure. And in the implicit condition, I explicitly tell them that they have to look for syllabic repetitions that occur very seldomly. So they are completely unaware of the fact that there is structure, and more or less I'm distracting their attention from the structure. I'm really asking them to track only syllabic repetitions. And what is beautiful is that it works with every single case that we observe tracking for both conditions, and of the same magnitude for the explicit as well as for the implicit. And here I'm also showing you the topography. So they are actually pretty similar as well. So it's not, so at least I think that for this very simplistic, low-level mechanism, it doesn't matter where someone wants to learn, they can just not help it but learn. Now, finally, so what is also interesting is that, remember that I show you first the evolution of the learning over time. But now if we actually parse those different responses over time, we have either the syllabic rate responses, which would be the blue. They don't change over time. The ones that are, at least in my view, extremely interesting are the red ones, which are the word rate responses only, that start at the very beginning, but then kind of die off, right? And the yellow ones, which are the syllabic rate plus word rate, that actually start, they increase over time, right? So it seems as if there is a, push between these two. And here are the topographies over time, so that you can see. So the first one that you see here, I hope, is that there's increase in the responses in the posterior STG, and then over time, these become far stronger, which are the word rate plus syllabic rate responses. And this, I think that loosely but interestingly resembles what Uri many years ago showed with Chris Honey, where they were scrambling images, or they were scrambling auditory stories in this case, right? And what they observed is that there seemed to be a hierarchy in terms of temporal windows. And one of their major findings is that in posterior stages, there is actually longer time windows as compared to A1, for instance, in this case, right? And this loosely resembles also what we find. So it could be that we are, having something similar to what they observed before. One very important note of caution, and I don't really understand this data very well, but I just thought I'd put it out here. So one of the problems with the statistical learning literature is that there's a lot of variability. Some people, have, higher than chance learning rates. Some others don't. And there's a big fuss about, people, have different learning rates, which I think is an interesting hypothesis. What we have observed is that whatever people measure offline may have nothing to do with what you measure online. So we measure really beautiful, and this is just one subject. So we have beautiful word tracking and zero behavioral output. So I'm not saying, maybe this is just because I'm measuring patients with epilepsy. Could be. Or maybe because we have a more sensitive measure. I don't know. But, I thought I'd just put it on at the discussion. So you could also argue that the results that we have maybe are just based on people that have epilepsy. So we did our job, and we repeated the experiment with, in this case, college students in the EEG, and we did the very same task as I showed you before. So either they were asked to explicitly look for structure or just implicitly look for the syllabic repetitions while they were listening to these materials. And what we observed was, again, that the explicit condition elicited the same word tracking response as the implicit condition, and we observed nothing in the random, but the syllabic tracking. Next. So I thought, if this is a chunking mechanism, shouldn't it work for other modalities? At least in my mind, it was like a simplistic idea. So if, and for all that we know in statistical learning, we can track higher order statistics, right? That's the beauty about these studies. And I borrowed the fractal stimuli from Nick Turk-Brown, and he has done beautiful research on this I'm going to show you later on. And with Nick, what we did was just we presented pairs of fractals in this case. So this fractal will always be preceded by this one, and this one in turn by that one, and so on and so forth. But the subjects didn't know that they also had structure, right? And we had a condition again that didn't have structure and one that did have structure. And so note that here the kind of like word rate or pair rate is the same, so it's 1.33 hertz. But of course, if you present images every 250 milliseconds, it's just too much. So we slowed that down. So we present only 2 images. So there's now duplets as compared to triplets that we had before. And the question was, would we get the very same segmentation signal? And sure enough, we do. And what is even more beautiful, at least, or I am particularly excited about this, is that we get again these, word pair or pair, only chunking responses that would not have any input response. And now notice that this is the input response, this 2.6, and this is the higher level response. And now what happens is that, of course, the yellow electrodes that I showed you before that were clustered in STG now are in early visual areas, which is what you would expect them to be, right? I mean, unless we got the anatomy wrong. Now, when I showed these results to Nick, he said, and Nick Turk-Brown, who has been working on that, he said, well, I have a theory. He said, I have the theory that the hippocampus is very important for these kind of things, and I'll tell you why. And his story goes that in the typical Magdalena 2 complementary learning theory, there is a dichotomy between learning that occurs in cortex that generalizes and episodic learning that occurs in the hippocampus. So the hippocampus would be good to store every single memory, like the dog that bite you, whereas the cortex would represent the story or would represent the fact that dogs are good, animals. That they exceptionally they bite you. Right? The problem here is that the hippocampus, while it's very fast learner, the cortex is not. So how could we support this learning in 2 minutes? It's kind of like a problem, right? So he came up with this interesting model saying that, okay, maybe this conundrum could be solved by thinking of the dissociation between the monosynaptic pathway, in this case that goes from the entorhinal straight to CA1, that could support statistical learning. So in this case, it would be fast, but would also generalize across experiences. Whereas the trisynaptic pathway would be holding the episodic memories. So we thought, okay, let's just look into that. And one of the interesting ideas that he pointed out to me that I thought was great for the kind of things that we're doing when we think of sequences and predictions and so on and so forth is that the hippocampus supports forward predictions by a pattern completion. And the idea here is very simple. So you pair two stimuli, and the first time, you present, like the stimuli, you have this elicits nothing in your brain. But the second time you present this one, this elicits a little bit this in your brain, and then as you repeat this more and more and more, the time you present the first stimuli, this evoked in your mind a forward prediction of what you will be seeing. And that's the idea of pattern completion. So there is something that comes to your mind because you present the first stimuli. And he and Nick Brown had beautiful evidence for this back then in the visual domain. What they did in this case, they in essence, I told you they borrowed his stimuli. So here you see that they borrow. So what he had done is that he had paired these stimuli, and he in the fMRI had first looked for what is the pattern that each of these stimuli elicit on its own, then had done the statistical learning sequence, and then he had measured again what is the activity that these patterns elicit in the brain after you have learning. And the idea is that if you have pattern completion, now by the time you present the first, you would somehow mingle it with the second. So therefore, you would somehow put these two memories together. Therefore, the separation between these two would become less, right? So the idea here is that after learning, after learning, now this pattern elicits that one, and therefore there is no differentiation between those anymore. And they look in this case at pattern similarity. The idea is that if they become more similar, then they are more confounded. And sure enough, they found that in the hippocampus as well as in the MTL, for valid pairs, there was stronger, mixed up of those memories. And so what we did now in this case was to look at, can we observe frequency tagging in the hippocampus as he would have predicted? And sure enough, we did very beautifully. So this is just one example electrode showing you the structure condition and the random condition. So there's nothing for the random. There's a beautiful peak for the structure. Furthermore, he had pointed out to us that the idea of pattern completion was important. And the idea here is that the moment you present the first syllable, it should be like an important one because it actually triggers the rest of the patterns. So therefore, the response to the first stimulus in the sequence should be different. And sure enough, that's the case as well. Right, so the blue one is the structure condition, the red one is the random condition. Furthermore, remember that I showed you that we had visual as well as auditory statistical learning? We thought, okay, one of the things that the hippocampus does is that it's a model. I mean, it shouldn't care whether I present, sequences in the visual domain, sequences in the auditory domain, should just, do the same, right? And it does, at least from, what we have observed so far. So we do have very beautiful frequency tracking in the hippocampus for auditory statistical learning as well as for visual statistical learning. So, so I hope that I was able to show you that the neural frequency tagging seemed to be a useful online measurement of learning, and I think that I think it's great to use it to unravel how the brain has structure, but also it's great to show how the brain can acquire structure. Right? Also, this is an interesting and puzzling result, is that there is an important dissociation between the offline measurements of learning and the online measurements of learning that we have. And this is something that I want to draw your attention to. So remember, we are using people that have epilepsy. What is one of the biggest complaints? So epilepsy usually comes from the middle temporal lobe. And people usually complain from memory deficits, right? So you would say, well, they shouldn't have anything of this, right? Unless they actually have preserved the monosynaptic pathway, which actually seems to be the case. It seems to be that the epilepsy specifically targets the trisynaptic pathway. So I think that, on the one hand, we can now use people that have epilepsy to try to understand these deficits, but also maybe it could be a useful therapeutic tool to restore memory in that population, just as a side effect. We weren't looking for that, and I think, and so this is the work of a lot of people. Nai and David are here. Nick, was, extremely important in developing these ideas. And Simon Henning, actually, he's the one driving everything. I'm just, presenting this stuff. And thank you.
Thanks, Lucia. Thanks a lot. Questions? Many. Good.
Thanks, Lucia, for bringing memory into this speech thing. It's really cool. I'm wondering, have you had a look at the directionality of coupling between the visual cortex and the hippocampus?
We did. I didn't show yet because we're still looking into the analysis. So it seems to be that the hippocampus is. I mean, I guess that you're going at is the hippocampus driving the responses in cortex, right?
Yeah. So the reason I'm asking this is because I think there was some recent data presented whereby the cortex itself can actually do pattern completion, especially in a statistical learning type scenario, which might not rely on single shot learning. Mm-hmm. So in this case, I think it is key to show whether the hippocampus is driving the pattern reinstatement in the visual cortex, or whether the hippocampus is reading out an already reinstated pattern in the visual cortex. Yeah. I think both scenarios could.
You're right. So from what we have done so far, it seems to be the former. So it seems to be that the hippocampus is driving cortex, right? But we are still working into that, and also you see, I mean, I didn't show you this, guys, because it's still preliminary, but we did direct cortical stimulation of the hippocampus, right? Because I think this is the best way to, if you shut down the hippocampus, then you shouldn't see it, right? And so far, this seems to be the case, right? But, I'm still not done with that yet, right? So it seems, that it seems to be hippocampally driven.
So you inhibit the hippocampus and the pattern completion in the cortex goes away? Right. Is that the thing?
Yeah.
That's pretty cool.
Lucia? Mm-hmm. Thanks for the great talk. So it might be a bit of a side question, but coming back to your idea of maybe using this effect, this statistical learning effect, to improve memory in patients with epilepsy, the fact that there's this dissociation between online and offline effects, how would that affect how epilepsy patients actually feel that they're improving?
So for once, and this is actually interesting, so what phenomenologically patients with epilepsy tells us is that because we play them first the random and then the structure, or the other way around, right? And sometimes we play first, for instance, the structure, and then they say, and then we play the random, and they say, oh, you know what, this is so fast. And it's the same stimulus. It hasn't, So I do think that there is something to the phenomenology. So once you are accessing higher order structures, things are feeling also differently to you. So I think this is something that we still need to, look more into. But it seems that they also have a time perception that changes as a function of, getting chunks.
Could it be a matter of just being more familiar with the material? Like if you would do it every day for hours, eventually it becomes an explicit knowledge. Like you have some meta knowledge about your own abilities.
I mean, you could do that, but you see, I think the explicit and the implicit are not the same thing. So we have tested also the explicit, and it doesn't. So we did it, say, spotting, which is there is structure, but you don't know what it is. Implicit, which you know nothing, and also explicit. So we tried to teach them the words, and so then they could actually follow them. What happens is that the last part is painful because they tend not to remember. So we have to do that a lot, and they get pretty frustrated. They tend to get the duplets more than the triplets, right? But what does work is that if you first, do the incidental for a long time and then you do the explicit, it tends to be easier, right? Than if you actually do it the other way around. So if you do the explicit, if you start away with the explicit, it's super painful, and they feel that this is super overwhelming, and it's not nice.
Joachim?
Thanks for a wonderful talk. This is also a little to the side. All of the frequencies that you were looking for were frequencies that are associated with the inputs that you're putting in there. And there are other reports in the literature that the process of learning itself can be associated with rhythms. For example, Earl Miller's work on learning categories involves coherence in beta rhythms between basal ganglia and prefrontal cortex. And are you going to show me something else?
No, I was just, so I said you're right.
So I was going to ask if you're seeing anything that's a frequency signature of the learning process itself, the way his beta was for the learning process itself. It goes away as soon as a a connection is made. You have something like that with words.
Right.
That is that.
No, I see what you mean. We actually haven't, for now we were dumb, because we thought, we have this frequency, so we just look for that. We haven't looked for any other frequency. But you're right that...
Do your data allow you to look at much higher frequencies
Sure.
like gamma and beta,
Sure.
which might be signatures that some learning is going on, and which will then go away as soon as things are learned?
Absolutely, yes. We haven't done it, but you're totally right. Yes.
Okay.
Yes.
Good.
Joachim?
Data. Thank you. So when I look at your coherence spectra, there are two possible mechanisms I could think about that would give rise to this word peak. One would be more temporally extended, maybe reflecting more like chunking or temporal integration, pattern completion. The other one would be more transient, maybe reflecting the transition probabilities. Would you know which one is true? Is it more temporally extended, more the chunking, or is it like basically reflecting the transient process, reflecting just the transition probabilities?
Yeah, so not now, but we're working on actually building a model where you can put, you can actually say, okay, what is the transitional probability? What is the strength of the model, right? What is the word frequency also? And what is the syllable? So we hope that eventually we will be able to answer that question, but I can't tell you the answer now.
Even if you maybe do like analysis in the time series domain, averaging on the onset of these words and then looking how that changes over time, maybe it would tell you if there's like a transient process or something more temporally extended.
So we have this kind of stuff. This is what you're thinking, right? Right? So we have the problem is that we see this in certain electrodes, in some others we see other forms, and this may relate to these kind of responses, right? So the word frequencies or the chunk frequencies come in different sorts, right? So you have either the chunk only or the chunk and the input, and those actually are different in terms of wave shape, right? And what I hope the model will tell us is whether these guys are the input to these guys, because in time that seems to be the case, right? This is what I was trying to make the case here, right? So in time, the chunk only guys kick in earlier, and then the chunk and input start to appear, right? So I hope, or I imagine that it could be like a baton passing, so something like that. I don't know yet.
Right. I'm going to ask you the same question I asked you Friday and see if I get the same answer.
Maybe not.
Maybe not. About why bother having a cortex? So 2 weeks ago, there was a paper, I think 2 or 3 weeks ago, was a paper by Tonegawa's lab about subsequent to learning, transferring the learned sequence or pair to cortex and then abandoning the hippocampal representation.
Right.
You're showing all these findings, which I happen to have seen before, that everything is done by the hippocampus, but Tonegawa's data suggests that yes, at the very beginning, but then you can throw it out. So I want to know why I need a cortex at all. Why isn't our entire brain just one giant hippocampus, since it can do all the work computationally?
I think that this relates to what this model is trying to put together, which is that there's a tension between actually storing the tokens, getting the general pattern, as well as an episodic trace of each and every individual. And the idea here is that you couldn't do this possibly only with the hippocampus. You have to go. So the idea is that the pattern completion goes to cortex, and it's cortex that tested it and sends it back, right? And that's the idea, that you strengthen the memory and get the token. So you construct the words.
But where are the tokens? I mean, the tokens are the episodic traces, right?
Right.
And the types are, let's say, cortical.
Right. And then the idea there, would be that, so if you take the input, say in this case, let me go back. So the hippocampus would be stupid. So then it would save something like 2p row, and it would also save the first time p row b and row b da, and so on and so forth. So it would actually store each and every of those, right? But that is dumb, because it won't help you generalizing over instances. It's only when you encounter these again, right, that actually that memory gets stored. And that's the idea of, like, those are the patterns that then would elicit higher pattern completion, right? And how do you store those patterns, at least in this account, is that you save that, generate a forward prediction of what you would have expected to hear, send that to cortex. If cortex says, okay, I found it, then you store the memory. But that would only happen in the monosynaptic pathway, not in the trisynaptic pathway, right? So it's the monosynaptic pathway that will actually be doing the generalization, right? So it's actually not, so it wouldn't be possible only with the hippocampus, right? But it starts in the hippocampus. That's why, like Simon was asking, like, okay, do you start in the cortex and then send it back to the hippocampus? Or in the hippocampus and then send it back to cortex, right?
Okay, because it's the same answer as Friday. It's good. It's self-consistent.
Other questions?
So maybe to relate to the question, I think the episodic memory doesn't work without the semantic memory, because if you don't know what anything means, you cannot build a memory with it. That is the idea for why we have concept neurons in the hippocampus. They're not there to tell us that Jennifer Aniston is a movie actor. They're there that we can build a memory in case we should meet Jennifer Aniston somewhere. So, and it goes back to the older memory work by this guy, I'm blanking the name on, the 30s. But whereby memory performance drops like a stone if you have meaningless stimuli. So I think the paradigm you have is super interesting, because it starts off as some random thing, then somehow meaning is extracted. And I think that a lot of memory theorists would agree that first you have to extract the meaning, and then you can build an actual memory, at least on a conscious level.
Can I ask you, Simon, what's the meaning of the pair? That's not more meaningful than the singleton. It's equally meaningless. It's just like the Miyashita, pairs of uninterpretable things.
And that's
what's the added
the
meaning?
case.
What's the semantic memory?
Well, I don't know, but you would have to form some kind of internal representation that tells the brain that this is something meaningful, more or less, however you interpret that. But if you don't have any meaning, you have a very, very, very hard time to build an episodic memory, right? I mean, that's just.
You see, I think that the point here, so I see from where you're coming, but I think that in this case, if you think of babies, they don't have meaning yet.
Exactly.
Right?
That's why.
So I think that the idea here is that you actually have to build, something that in this case is, it doesn't have meaning. So even if you actually get the structure, it doesn't have meaning. It only gives you like a chunk. It tells you like, okay, this is a piece.
Yeah.
Whether that piece is important, relevant, has a meaning or not is something that you can do later on. But this is just a chunking mechanism. So
what I think could be going on here, but that's something you can test, is that first the cortex pulls out some statistical regularities, I think. And then the hippocampus somehow picks that up. And then at that point you can form association. That is what I mean with meaning here, that you pick up some regularities. I mean, obviously there is no semantic meaning to that, but some sort of regularities. So the cortex slowly learns these, right? And once it's learned that, it can do some one-shot learning in the hippocampus, maybe.
Yeah, we think that. So what I can tell you is that the cortex is very good to learn stuff like mismatch negativity. So ta 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 pa, that is really good, but it's more like adaptation-like. This is a different mechanism, right? Because there's nothing that repeats. So you really have to build structure. And these I haven't seen very quickly in cortex, right? Or put it differently, I have seen it in hippocampus earlier than in cortex, right?
So what does the behavioral data tell you here? Do the patients consciously pick up on that when you ask them?
No. So you ask them, and then they say like, look, right. I mean, both of them look gibberish.
So it's all subconscious. It's all implicit learning that we're talking here.
Absolutely. And they all think that they are guessing,
Okay.
And actually, each and every of the patients that I've tested so far, they say like, look, I did terribly. And in fact, behaviorally they do, not neurally. So it's kind of like a funny.
So that might be the issue here, because episodic memory is obviously a conscious form of memory, right? And what you are presenting here is priming implicit learning in the hippocampus, which, puzzling.
I know.
Well, I wanted to bring the name of Anselmo here. He was looking at grid cell structures in the hippocampus and related it to theta and face tracking and all that. And I wonder if somehow this mechanism can be used, for what you do.
Most likely. So I discussed this, results with, I think I told you, with Matt Wilson, because he does have the theory, in essence, every time you have like time-varying inhibition, that brings you an integration window. And he would say that the hippocampus does integration over very short instances of memories, like this sort of, like 2 or 3 syllables. And then say the precuneus and, frontal would actually do longer stretches, right? But the idea is that it replicates the same mechanism. So in that case, you would, you could think of this in the same way as theta phase precession. It's not very different.
Very well. If there are no other questions, one question.
I'm connecting what you said beautifully with the random cloud sounds we heard about this morning and the immediate learning of the adjacent repetition, and whether it would be useful to disrupt the sequence and have immediate repetitions so that that pattern is immediately learned, and so you get even stronger effects. And have you tried that, and what are your thoughts?
You do,
about
yeah.
that?
But you see, so the problem is repetition. For some reason, the brain is very drawn to repetitions, and they draw attention. So it's very hard to know whether you're learning the structure because you pay attention, or because of the mechanism that underlies the repetition. You see, and Nick actually
but
has
how
played.
what do you mean you pay attention? You pay attention?
No, they just draw attention to you. Like every time you have a repetition, this is work that Nick has done, before in statistical learning. A way to discover very quickly the regularities is by actually repeating them. But now what they found is that a confound there is that every time you do something like 2pp, like somehow the brain likes the repetitions, and therefore it pulls attention towards those. So then you don't know whether it's because you learned the statistics or this attention. Right?
Yeah, so just a quick question. So there's plenty of models on hippocampal sequence learning based on LCP, right? So if B is presented after A, you get strengthening, but otherwise not. So I wonder if you take some of these models and then expose your sequences to those, whether you will see this dynamics emerge.
I mean, that's an excellent. I haven't done it, but I think that these are all the things that we should be doing. I mean, in essence, what I want to draw attention to now is that the hippocampus may be doing something as well, not just cortex, right? That's all what I'm trying to say. So.
Thank you, Lucia.